Alzheimer's disease is one of the most common age-related neurologic diseases. It involves degenerative changes and cell death in the cortex of the brain, with gradual loss of cognitive function. Many factors contribute to the disease, but the fundamental process is age-related epigenetic changes in the glial cells of the brain. Neurons depend on the glial cells to function normally. Microglia, one type of glial cell, constantly produce and absorb molecules in a continual recycling process. One common example, beta amyloid proteins, are produced and secreted by the microglia. Some of these proteins are sticky and apt to form clumps. But they are usually reabsorbed back and broken down. This molecular turnover is rapid in young cells, but as the cells age, molecular turnover slows down, leading to a gradual accumulation of the microaggregates around the cells. As molecular turnover slows down even further, the microaggregates grow larger, forming beta amyloid plaques and damaging the neurons. Over the years, these plaques increase, as does the damage to the neurons, which begin to fail and die. Molecular turnover slows down because of subtle but pervasive changes in gene expression in aging cells, which are the result of telomere shortening. Telomeres are the regions at the end of each of our chromosomes that not only protect the genes, but also serve as aging clocks, shortening over time. Over the human lifetime, cells may divide many times, and each time the cell replicates its chromosomes. The replication protein complex attaches to the end of the DNA chain, but can't read the tiny segment of telomere that lies beneath its attached area. So, with each replication cycle, the telomere grows slightly shorter. And as the telomere shortens, it affects the rate and pattern of gene expression throughout the entire chromosome. As our cells divide, telomeres shorten, gene expression changes, protein turnover slows, and cells fail and die. The final result is Alzheimer's disease.
Welcome to another episode at the Rise of the Ageless Starman. Today I have the privilege to host Dr. Micheal Fossel, the driving force behind Telocyte. Micheal has been the leader in proposing the use of telomerase to treat human diseases for the past 2 decades. After he completed his PhD in neurobiology at Stanford University in 1978, he went on to finish his MD at Stanford Medical School in 2.5 years. Dr. Fossel was awarded as the National Science Foundation Fellowship and taught at Stanford University, where he began studying aging, emphasizing premature aging syndromes. Dr. Fossel was a clinical professor of medicine at Michigan State University for almost 3 decades and taught the biology of aging at Grand Valley State University. In 1996, Dr. Fossel published Reversing Human Aging, the first book to ever describe the medical aspects of extending human telomeres and the potential for curing age-related diseases. His recent book, The Telomerase Revolution, discussing aging clinical disease and the prospective FDA clinical trials of telomerase therapy. It has been praised in full-page reviews in London Times and The Australian, as well as being named as one of the most 5 science book of the year by the Wall Street Journal at 2015. He is the medical advisor for the Dementia Society of America and has been member of numerous scientific organizations, including the American Association for the Advancement of Science, the American Aging Association, the American Gerontological Society, the American Geriatric Society, and the Alzheimer's Association. His book, Cells, Aging and Human Diseases, is the first textbook to explore aging all the way from genes to clinical application, analyzing the fundamental cellular changes which underlie human age-related diseases. He has a personal experience with a number of biotech companies both as inside or angel investor. Geron, Sierra Science, Phoenix Bio, Molecular, and more. This is just on a nutshell of Dr. Fossel's rich career. Stay tuned as he will explain us about his current project with Telocyte and how they target to cure Alzheimer and other age-related disease with telomerase gene therapy.
We're on.
Okay. Hi, Dr. Micheal Fossel. Thank you for joining to my podcast, The Rise of the Ageless Starman. Before we start, I want to acknowledge you for being an inspiration and your stubbornness to try new creative ways to deal with Alzheimer. Even though the main the main concepts are a little bit I know it for you it was hard to push your concept. You will describe it later, but first of all I want to thank you very much and your passion and your patience to keep teaching everyone, even though you're now in your industrial career. Thank you very much.
Bavakasha.
So, like first question for you is, what is the difference in your approach from other companies to treat Alzheimer?
I think the main difference is that for decades, centuries, in some respects, we have all been the prisoners of our assumptions. The best analogy I can think of is the Middle Ages, when no one knew anything about microbes. They thought everything should be treated with herbs. And they can be very effective for symptoms, but if you're trying to treat, for example, smallpox or plague, you need to understand microbial disease. And I think that's where we are with regard to Alzheimer's and other age-related diseases. We are prisoners of our assumptions. We assume that what we're looking at is the mechanism without looking further down and trying to understand the fundamentals of the disease, as again, prior to the onset of our understanding of microbial disease. It's that assumption that we already understand the disease that blocks progress. So from our perspective, what we are doing is looking at more fundamental mechanisms and asking, why do age-related diseases occur, and specifically the dementias, and more specifically, Alzheimer's.
Okay, and you, like, with your company Telocyte, that you are trying to, that you are the founder, are you over with the animal studies? Can you explain how you conduct them, the studies?
No, there have been a lot of animal studies over the past few years, but we are required to do what's called an animal toxicity study. And we're going to be doing that in dogs under FDA auspices. And what that is a study that gives the FDA confidence that we can do this safely in humans. So we need to meet that obstacle before we can move on to human trials.
Okay. And how much money you needs to be invested in order, like, to move to phase 1 and 2 in clinical trials, do you think?
More than I have personally.
Yeah.
On the other hand, it takes, let me put it this way, I think it takes a lot less money to do something effectively if you aim at the right target than it does. For example, one of the major worldwide pharmaceutical firms has spent almost $1 billion on a monoclonal antibody set of studies over the past few years and been unable to demonstrate any efficacy. We can go through a phase 1 trial for something a bit in excess of 10 million US dollars, and for an additional 20, finish the phase 2 trial. And what that I think will do is allow us to demonstrate that our approach actually works. And that's certainly not something you can say for spending $1 billion on programs that don't work. So yes, relatively cheap, more than I certainly have available myself. But from a pharmacological perspective or a pharmaceutical perspective, it's a very cheap investment if you aim at the right target.
Okay. And in recent years, like let's talk about Telocyte's team. You, a few members joined you. How is it affecting the company, and what did it inspire them to join you? I know they are big shots in their
Well,
field.
right. We have gone out of our way. Let me preface this by saying our major concern is not just the cost and the timing of this, but the credibility of it. We do not want to make a misstep. Because when you think about it, depending how you define them, there have been more than 1,100 interventional trials aimed at Alzheimer's. And by global consensus, none of those trials have demonstrated the ability to change the disease course. We think we can not only slow it, not just stop it, but actually reverse some of the cognitive decline. Now, to make that statement, we're going to have to make that statement and back it up. We're going to have data. But even if our trials show that, we recognize that most people, sensibly enough, will discount that result. So we have to be very careful in what we do. And that's been true of the way we approach the FDA, the way we deal with our advisors, the kind of talks we give, where we go out in public. But it's also true of our scientific advisors and our clinical advisors. So, for example, we have gone out of our way not to pick people simply because they have big names, but more importantly, people who have respect, credibility globally. So for example, on our scientific advisory board is the editor-in-chief of the world's most prestigious Alzheimer's journal, who asked to join us, and I'm delighted he did. We also have probably the world's most important animal study group. It's a company out of Canada, and their CEO has joined us. The woman who is behind the design for many of the monoclonal antibody studies worldwide for Big Pharma and for the design of their statistical work has joined us. So we've been looking for people who not only have credibility, but they really have hands-on experience. These are people who get their hands dirty doing things, not just having an academic credential, but in fact, they have accomplished things, either running a business or running clinical trials. The same has been true of our clinical advisory board. We picked people who have been running Alzheimer's trials and who are very skeptical. And I have said to each of them in turn, we are not interested in people who drink the Kool-Aid. That is, people who are true believers in our model. We're looking for people who are skeptical, who are intrigued by our model, see the potential of it, and want to see it rigorously tested. We don't want to make a misstep. And as I say, when we come out the other end, I expect that in our first trial, we may be able to demonstrate clinical improvement. But to have that be believable, we have to be very careful what we do.
And yeah, so a little bit about the technology, the activation part of the telomerase. Can you explain a little bit of it? How it goes?
Yeah, we're not just activating telomerase, we're actually putting in a telomerase gene that's transiently expressed that will reset telomere lengths. And I have to say that telomeres are not the cause of aging or disease. However, they are probably the most effective point of intervention for aging or disease. It sort of reminds me of the whole discussion going on with COVID right now. I think we have a fairly good understanding of the virus, but the question is, where's the most effective point of intervention for a vaccine? And that's why some of the vaccines vary in their ability. The same thing's true here. There are lots of places we might intervene with Alzheimer's or other diseases. The question is, where's the optimal point of intervention? And what we're doing actually is using a telomerase to re-extend telomeres and using that to reset the pattern of gene expression. So for example, the neurons and glial cells in your brain, we are trying to get them to act more like a 40-year-old brain than a 70-year-old brain. I think that's a good summary of it.
Yeah, and like usually when the discussion is about the telomeres, it's about their length and how it keeps the rest of the DNA function. This is a basic understanding of mine of the problem.
Anything you could say and anything I could say is a simplification. You know that, I know that too. Let me start by saying that telomere length is completely irrelevant. What matters is the change in telomere length. So for example, there are varieties of mice that have telomeres that at birth are 10 times longer than mine, and yet I have a lifespan 40 times longer than theirs. And the reason is it's not the telomere length, it's the rate at which telomere length changes. In fact, you can predict species lifespan based on the rate of change. And even there, it's not the change in length per se that's the question, it's the change in gene expression or the pattern of gene expression. So an analogy I sometimes use is a symphony orchestra. If I have a symphony orchestra that one night is playing in atonal composition, for example, John Cage, and the next night is playing Mozart, the difference is not in the instrument or the first violinist or the oboe player or the tuning of the piano. The difference is the score and the tempo. So the same is true of the function of my for example, microglial cells in my brain. The difference between my microglia at age 7, right now I turned 70. My microglia, what's that? My birthday is tomorrow. I turn 70 tomorrow. But the difference is the pattern of gene expression. So if I look at the way those genes are being expressed, the same genes that I had when I was 7 years old, the difference is they are playing a different tune. Same instruments, same genes, different tune, different epigenetic expression.
Yeah. Well, actually, my first guest said exactly that, even though it wasn't only in telomeres, but about the rate of the pace that it gets shorter.
Let me give you an analogy. Telomeres no more cause aging than genes cause evolution. You know, genes aren't a cause of evolution, but they are a key player. If you want to understand how evolution works, you need to understand genes, but they're not the cause. That's a misnomer. The same thing is true of telomeres. Telomeres don't cause aging. They're simply a central player in aging. You need to understand telomere mechanics to understand how aging works, but to talk about causation is sort of simplistic. It's too complicated a system to sum up with a single sentence about causation. It's much more complicated than just A causes B.
Yeah, so you are pointing to the microgalia cells, and what about mutation in the DNA inside the microgalia cells?
Mutation?
Yeah, can this cause Alzheimer too? Not only the rate of the shortness of the telomeres?
Interestingly enough, again, the same answer. It's much more complicated than sort of yes or no. So let me give you a couple of examples. First, let's back up and take a different allele, a mutation, if you will, but a different allele. Let's say that I have 2 ApoE4 genes, which means I have a high risk for Alzheimer's, or I have 2 ApoE2 alleles, which means I have fairly low risk. And what you find is that neither one of those causes Alzheimer's, but what happens is if I have 2 ApoE4 alleles, the beta amyloid I make is a slightly stickier molecule, so it tends to aggregate faster. But you don't really see that as a problem until you slow down the rate of turnover of the microglial turnover of beta amyloid. And if I have 2 ApoE2 alleles, I have very low risk because again, the molecules don't tend to be sticky. So even at age 90, I may not have any microaggregates, whereas if I have 2 ApoE4 alleles, I might have microaggregates forming at age 40. But it's the rate of turnover of the beta amyloid that's the key factor here. And yet, those different alleles make a difference. Now, the same thing is true of if you look at mutational problems. But there's an interesting wrinkle here, which is that as the epigenetic expression of your cells change, for example, in microglia, what you find is the rate of turnover, recycling, if you will, the rate of turnover of molecules changes, not only collagen and elastin in my skin, but for example, beta amyloid or tau proteins in my brain, but also other molecules that are critical, for example, DNA repair enzymes. There are, roughly speaking, 4 families of DNA repair enzymes. One recognizes it, one cuts it out, one puts in the new base, and one sort of anneals it into place, or these families do. But all 4 of those families, their rate of recycling goes down with cell aging, cell senescence. So what you see is that as cells get older, their ability to repair DNA goes down in terms of the rapidity which they do it. So for example, since you and I started this conversation, probably every single cell in your body has had some DNA damage, and you have probably repaired it flawlessly. But the rate at which you repair that goes down with age, which is why you see an exponential increase in the rate of mutations, failure of DNA repair, as we get older. And that is independent of years, its lifespan. So for example, a mouse has an exponential increase in the rate of mutation over 2 years, whereas you and I have that same exponential increase over, say, 80 years. So cell senescence and telomeres and cell aging and aging in general play an enormous role in the rate of mutation in cancer. You might, in fact, say that the cancer comes in a couple of different varieties. You know, there are those that you sort of inherit as sort of bad genes at birth. And here you might think of a BRCA gene. But there are also this rate of DNA repair that goes up exponentially with age. So there's an early form of cancer that kids tend to get, and there are later cancers that most of us will tend to get. But you might also say that the rate of cancer as we get older is not, strictly speaking, due to exposure to carcinogens. It's the rate at which we repair the damage as a result of that exposure to carcinogens. And that's age related. So all of this plays into one big complicated system. I think I understand it more than most people do, but I don't understand 1% of what really is going on. It's so complicated, it's just astounding.
Yeah, actually, my last guest, she had Debbie Teuber from Ben Gurion University, is dealing with SIRT6. We had a conversation about it, about the damage of the DNA and how SIRT6 can identify it and repair it, and how it declines with age. Mm-hmm. I invite you to listen.
Yeah, it's interesting because, you know, if you look at those four families, as I say, there's four major families, but each one goes down on the rate of turnover. So your likelihood of identifying a DNA error is slower as you get older. The rate at which you replace it is slower, or cut it out is slower. The rate at which you replace it is slower, and the rate at which you ligate it into place is slower. Those are multiplicative. So 1 multiplies by 2, multiplies by 3, multiplies by 4, which is why there's this exponential increase in the rate of cancer with age. It's expected.
Yeah. Let's go again back to the technology, like how, like Telocyte want to do it with injection. Yeah, one injection.
Yes. Now, you know, it's effectively what we're trying to do is reset the cellular clock. And if that's true, then we'll need to reset it again. Our best guess is someplace between every 3 and 10 years, you'll need to be treated again, but the initial treatment is just once.
Okay, so how do you know it will, how will one single injection telomerase therapy will be targeted only to affect the CNS, and does it have any side effects what are the risks that you are afraid of?
I wouldn't say I'm afraid of the risks, but I'm aware of potential risk. Oddly enough, most of the side effects tend to be benefits. What?
I say we don't have nothing to lose here.
Well, that's part of the reason we're going after Alzheimer's. You know, the risk-benefit ratio is enormously in our favor. You know, if I were trying to treat osteoporosis, people don't die directly from osteoporosis. You may fall at age 70, break a hip, go into surgery, get an infection, and ultimately die. But osteoporosis per se is not fatal, but Alzheimer's is. And furthermore, with osteoporosis, there are multiple other therapies that people try, biphosphonates, calcium, hormones, with various degrees of efficacy. But with Alzheimer's, there really is no alternative. So we've got a fatal disease with no therapy. And so the risk people are willing to assume, and likewise regulatory bureaus like the EMA or in the US, the FDA, it's a better risk-benefit ratio. Patients are more likely to try something because, again, what options do we have? But the risks, there are a number of possible risks. I mean, the obvious ones would be just sort of generic risks in the body. And what you find is that based on the studies we've seen, most of the risks turn out to be benefits. But a couple of specific risks. One would be cancer. And for 25 years now, there's been a concern about telomerase in cancer. And again, it's far more complicated than people think it is. It turns out that the longer your telomeres are, the longer the telomeres are, the lower your risk of cancer. Except when, it's not a straight curve. It tends to kind of go up, peak, and come down. That is, your highest risk of cancer is to have fairly short, but not zero length telomeres. because if you have almost no telomeres, most cells won't divide in the first place. So you don't get a clinical cancer. You have a malignancy, but it's a single cell, for example, unless they do an end run using an alt mechanism. But the upshot is that your lowest risk of cancer is when you're young and your telomeres are long. And that's the kind of thing that we see if you look carefully at the data. It's a very complicated relationship. It's not linear at all. So,
wait, I didn't hear you. There was a disconnection.
I saw that for a minute. So there's a very low risk of cancer when your telomeres are long.
Yeah.
Mostly because you're repairing your DNA much faster.
Okay.
Now,
Okay.
the other risk, want me to go on, Gil?
There was a disconnection.
Yeah, I'm sorry.
So after the point, you said there is a point of optimum telomeres length.
Would it help if I pulled up a graph of that?
Yeah.
I can do that. Just if I can spot that for you. This will be taken from an article of mine. Oh, here it is. Yeah, this is sort of what it looks like.
Yep.
What you're seeing is that these are telomeres as they shorten. And this is your risk of cancer that goes up and down. And this is your risk of mutation. And what you find is as telomeres shorten, your risk of mutation goes up, but your risk of cancer peaks and then falls because here your cells have very little damage, so you're not likely to have cancer. Here, you're very likely to have DNA damage and your cells can still divide. But if your telomeres are short enough, most cancer cells won't divide. Again, there's an escape route for this, an alt mechanism. But this is your peak risk in here. And if you re-extend telomeres, what you expect to find is less cancer. And in fact, that's what we find in the animal studies. So yeah, it's a misunderstanding to think that increasing telomeres or telomerase causes cancer. It's a lot more complicated than that. Let me put myself back to my background there.
Yeah. Okay. So was there any something similar, chemical treatment that was used on humans or on animals?
You mean in regard to telomerase?
Yeah.
Yeah, there've been it's actually interesting. If you look back historically and you ask, will we ever cure aging? In fact, technically speaking, we first did this 22 years ago with the first studies showing that you can actually reverse aging in human cells in the laboratory. And then we first showed you could do this in human tissues 20 years ago this year, starting with skin cells and moving on to arterial cells and then bone cells. But then there were some interesting studies that you're sort of alluding to, which is looking at telomerase activators. In this case, estragenol is probably the most active of them, commercially available at least. And what you find is there have been, I think, at least 4 reviewed studies. In fact, I published some of them in a journal I was the editor of for a while, looking at this. And the data is intriguing. It's not overwhelming. You certainly didn't find that if you use these telomerase activators, you reset from age 70 to age 30. But you began to find a number of physiologic biomarkers that were improved. For example, response to glucose. For example, T cell response to stress. For example, bone mineral density. A lot of things you look at, you find that they improve with telomerase activators. They are not nearly as effective as you would like it to be. And again, the data is not overwhelming, it's just intriguing.
Yeah, so it might be a concentration problem, like that if you will increase the dose, it will give you more overwhelming results?
Probably not. There was at least one study on that suggested that if you increase the dose, you got less effect. But that was a small study, and there's a lot we just don't know about it, frankly, Gil.
Yeah, okay.
In fact, I'd have to say we don't know a lot about almost any of this in the long run. This is early. All of this is very early in the field of understanding aging and age-related diseases. People don't realize what we're about to do, but it's early stage. It reminds me many times of sort of the first powered flight at Kitty Hawk with the Wright brothers. Yes, they got it up in the air, But no, that was not an Airbus 380. It was early days.
Yeah. Well, that's
And yet that was powered flight. It was done. I think that's where we are with regard to this sort of therapy. I think we're going to demonstrate we can do something that's never been done in history before, but this is still early in this whole field.
yeah. Okay. And who are your partners? Like, I know you have some partners that helps you with the technology.
Yes, I'm not sure which ones you're thinking of.
Yeah.
I mean, as I say, we've got a number of people on the scientific advisory board who are very good at this. We've also brought on one of the postdocs who actually did the original work with Maria Blasco in Madrid at CNIO. He's on it. We have a contract research organization that's probably the world's best to help us with this. We've been in touch with a number of the gene therapy vendors that we'll need to use. And again, we've been talking to people like Lonza and Wuxi who run these things globally. And we have recently brought on a consultant who may join us, who's been taking a lot of these gene therapy trials to the FDA, and we're delighted to have her. We originally had the chief scientific officer for Avexis, who is that first person who put through the spinal muscular atrophy study, both in New England Journal of Medicine, and then brought it through for a Zolgensma study with the FDA, and that's been released as gene therapy. Now, he had some issues with the FDA, and so we're not officially using him anymore. But we've concentrated on people who have actually done these things, not just people who talk about it, but people who really know what they're doing.
Yeah. Okay. So I know, as we said, it's only the beginning of this science.
Very much.
But still, what else do you expect to see scientifically in the results, besides like turning Alzheimer background? What you have expectation to see? What?
Yes, this is what's generally called a platform technology, which is to say it's not aimed at a single disease. It's got implications for a number of diseases. In this case, anything that's age-related. There are, for example, if we're just taking the central nervous system, we're aiming initially at Alzheimer's, but we would be happy to go after Parkinson's or any other age-related dementia. And there are some interesting clues that ALS, amyotrophic lateral sclerosis, may also be a good target for us. And anything else, again, that's an age-related disease, anything that seems to increase with age. And after we're dealing with CNS, our next target after that is going to be age-related vascular disease. That is strokes, heart attacks, aneurysms, peripheral vascular disease, congestive heart failure, the list goes on and on. Those are actually, by percentage, the biggest killers in the world currently. And after that, we'll be looking at other age-related disease, for example, podocytes in chronic renal disease, pulmonary fibrosis, COPD, osteoarthritis, osteoporosis, and so on.
So the potential is huge.
Yes, it is. If I look back historically and I say, what is the most important thing medicine has ever done to make people's lives better? It is not incremental technical advances like robot surgery or heart transplants or the latest statin. Those have a value, and they often are expensive. But if you look back historically, back about 200 years, I think it was the advent of the concept of microbial disease that had a bigger impact on human quality of human life than anything else. The idea of washing hands, for example, before you deliver a baby, drastically cut both maternal and neonatal death rates. The concept of immunization, sterile surgeries, so on. First polio vaccine, the first rabies vaccine. These made our lives better. And interestingly enough, they actually lowered the cost of medical care dramatically in many cases. And I think that we're about to do the same thing with regard to age-related diseases. If I go back to 1950, there were a lot of people concerned in various nations, for example, the UK, the US, and others, about the cost of treating polio. And there were people who actually estimated that the US, for example, would be medically bankrupt by the year 2000 because of the cost of treating polio victims, because of iron lungs, braces, rehabilitation therapy, nursing care, pulmonary problems. And yet, globally right now, the cost of treating polio is about 10 cents per capita of treated patients. I think we're about to do the same thing with regard to age-related diseases. So for example, A number of nations right now are concerned about the cost of Alzheimer's disease. And in the US, as an example, typically the last year of life is $100,000 US or more to treat a patient in the nursing home in the last year of life with Alzheimer's. I think we can cut the cost of treating Alzheimer's from in excess of $100,000 a year to one $5,000 treatment that effectively cures Alzheimer's. In short, I think we can entirely obviate the concern about long-term costs globally, nationally, and personally for age-related disease
Well,
and improve human lives. Thank you.
yeah, that's why we're here.
I think so. Again, I'm not impressed. I'm glad we have technical advances. Don't get me wrong. The latest statin can save some lives. But I think we do much better if we can understand our assumptions about how disease work, dig a little deeper, and the more we understand how these work, the better off we are. From our perspective, I would say that the major problem we've been having, it reminds me of the Indiana Jones movie, where Sala and Indiana Jones are talking about how the people are digging in the wrong place. We are often digging in the wrong place. And you cannot expect to find a cure for age-related disease if you keep putting money and digging in the wrong place.
Yeah, and like it's a complex system, so I don't know why to concentrate only in one basket, all the balls.
Well, again, I think the problem is we're a prisoner of our assumptions. We assume we understand something and we don't. Lord Kelvin, for example, in 1895 made the statement that heavier than air aircraft are impossible. And he was not ignorant. He was not superstitious. He was not naive or foolish, but he had assumptions about aerodynamics that were flawed. He understood about weight. He understood about air pressure, but he was a little weak on aerodynamics. And he assumed he understood the problem, but he didn't. And that's an issue that we always have. In the early 1900s, we assumed that we understood what atoms and subatomic particles must be. They must be little teeny particles, and they're not. They are quantum things. And it wasn't until 1924 in the first publication, and that we began to get a glimmer that our assumptions about atoms being billiard balls and protons being smaller billiard balls were flawed. It's the assumption that we understand it that gets in our way. And I think that's what happens with aging. People tend to assume that aging, quote, just happens. It's just entropy. No, it's a lot more complicated than being just entropy.
And I think in Alzheimer's specifically, with the beta amyloid conception, it looks too good to be true. But it looks very simple and logical that if we have matter that destroys the neurons, if only we can take it out, it will everything will be okay.
Well, here's an analogy for that. Yeah, we've known about beta amyloid for in excess of 110 years since Alois Alzheimer's first described it, and even he warned against people thinking of that as the cause. And more recently, it's been tau tangles and then mitochondrial dysfunction and microglial activation, and the list goes on and on. But we could make the same mistake by looking at Ebola. We could say Ebola is no more than a bleeding predilection, a bleeding diathesis, plus nausea, plus fever, plus white count, plus aches and pains. But that's not what Ebola virus is or Ebola infections are. It is a complicated interaction between a virus and a very complicated mammalian organism, namely you and I. It is not simply a collection of symptoms. And if you treat the symptoms, for example, by giving blood products and pressure agents and fluids and antiemetics, that may help, but it does not treat the disease. And I think that's where we are with regard to Alzheimer's. If we just treat beta amyloid and tau tangles, you may or may not get some borderline effects, but you're not treating the problem.
Yeah. And like one new assumption is that you can this is a new approach, relatively new approach, suggesting that restoring the vitality of the immune cells will possibly regain the cognitive abilities and will encourage waste disposal and manage to protect neurons from death. This will lead to a better function of the microgalia cells and less delay Alzheimer, possibly. You think it can go backwards like that? If we treat the microgalia and the brain will function better, the body will have less damage, less
Yes
disease?
and no. I mean, the problem, Gil, is that people tend to look at this in a component way. Sometimes the component they look at is beta amyloid, or the component is tau protein. But they do the same thing if they think it's just a matter of immune function, or just microglia, or just astrocytes, or just anything, the vascular endothelial cells. They're still looking at it component. In that case, they're looking at it in terms of the histology component rather than the chemical component. But it's more than a component problem. It's a broader problem than that. So if we only treat, for example, macrophages, or we only treat astrocytes, or we only treat T cells, or it's still a problem where people are missing the point. They're not going deep enough and saying, this is a complicated system in which it's more than just a component. Here's an analogy of that. I have a friend who, in fact, our CEO, was responsible for overseeing the design of some of the Rolls-Royce jet engines. And if I have a fan blade in the Trent 800 turbofan of that engine and it fails, the question you should ask is not why did that fan blade fail, but why did that fan blade fail in the turbofan, in the Rolls-Royce jet engine, in the Airbus 380 at 35,000 feet, at hundreds of miles an hour, going hundreds of RPM at a very high temperature. In short, it's a system problem, not a fan blade problem. And if you just look at the fan blade, or you just look at any particular component, you're missing the problem. The problem is systemic. It's an issue that's dynamic, not static. And that's what's going on here too. If you just focus on T cells, or just focus on immune function, or just focus on any particular cell or any particular physiologic process, you're sort of missing the system. You're missing the dynamic portion of what's going on with this cascade of pathology in age-related disease, and particularly in Alzheimer's.
How do you think like computer power will help you? You said it's a system.
Yes, but not much. I mean, the computer power won't help much. Let me give you an analogy too. Let's say that you and I, again, went back to 1500s, early 1500s in Europe. And again, we know that there's a smallpox epidemic coming. And all of the local herbalists are extremely bright people and extremely knowledgeable, more so than I ever will be about herbal medicine. And seriously, they knew a great deal. But they have this conception that things can be treated with small molecules, that is herbal medicine. And they had no concept of microbial disease, invisible little creatures that attack you, and they wouldn't believe it. Now, let's say we could go back and give them access to quantum computing, massive data crunching, artificial intelligence, enormous supercomputers. The problem is they'd still be asking the wrong question. They try to identify which is the right herbal medicine and what doses and what combinations to give, but it's the wrong question. And the same thing is the danger here with regard to trying to deal with Alzheimer's and artificial intelligence or quantum computers or anything you want to throw at it. It's not simply a matter of identifying the right small molecule. It's a matter of understanding the system itself. And it's the danger of your assumptions. You're a prisoner of your assumptions. And if your assumptions are, for example, that microbes don't exist, you'll look in the wrong place. If your assumption is that all of Alzheimer's can be caused by beta amyloid and 14 other small molecular problems that can be dealt with small molecules, and you attack that with artificial intelligence, you're still asking the wrong question. You need to step back and understand your assumptions. Another example might be the early Vatican astronomers. Back again, 500 years ago, were trying to calculate the orbits of the planets, but they started with the assumption that the Earth was the center of the universe. So they got these increasingly complicated epicycles. And if they had had access to quantum computers and artificial intelligence, they would have calculated precisely the location of the planets, but their entire assumption was wrong in the first place. The premise was wrong when they started. So since they asked the wrong question, they get the wrong answer. Very good calculations of where Mars will be in 43 years and 2 days. But they don't understand the system itself. That's where we are with regard to age-related disease, Gil.
Yeah. Well,
I like computers. I like AI. I'm in favor of it. But you have to understand that the limits are imposed here, not there.
but maybe, like, again, what I think that the problem with AI regarding to biology,
What?
that it's hard like to do clinical trials on humans, and the data, well, there are like AI is supposed like to ask questions that uh, we couldn't think of in the beginning from the data itself, but with the medical development, the data itself is not on humans, so we are.
I know a lot of people are trying to talk about, instead of in vivo and in vitro and ex vivo experiments, talking about in silico experiments where you can run these things essentially by modeling them. And again, there's an enormous benefit to that as long as you watch your assumptions. Those assumptions can destroy your entire conclusion.
Yeah, from your understanding, um, like we know that cells divide and everything gets each time they divide it, the process is we are getting older. This is, you think there is something in a lower level that causes the aging process?
I'm not sure what you mean by lower level.
Like
The answer probably is yes, but I need to understand which direction is lower.
like is there a process that is parallel to this that causes like are there?
Let me use an analogy here. Again, one of the things that used to bother me 50 years ago, sort of in graduate school, was that people always would shrug when it came to aging and say it just happens. Things rust, they fall apart. What do you expect? And let's take two houses. Okay, here's my house behind me. Take two houses, both built in the same year, both built with the same exact construction. Everything is the same. And one house, the people live in it take care of it. And they repaint it, and they take care of the windows. They replace missing pieces. They take care of it very carefully every year. The other, they just let it go. They don't worry about it. And I go 150 years forward in the future. This house is still doing very well. This one is not. The entropy has been precisely the same, but the maintenance here has been superb. The maintenance here has been non-existent. And the same is true of a garden. I take two gardens, and one of them I weed and I care for. The other I ignore. This one, 2 years from now, is hard to find. This one looks gorgeous. It is a matter not of entropy or weeds or anything else. It's a matter of maintenance. In biology, we talk about red queen theory. And here the idea is any system, for example, a human body, is continually being threatened by, for example, bacterial infections, viral infections, predators, you name it. It's part of biology. And the only way to sort of stay in one place is to run very fast. That is, my immune system has to be able to keep up with the latest COVID virus. Actually, I'm probably one of the only people you've met has actually had COVID vaccine. I think I'm safe. But you see what I mean? It's not a matter of the threats that kill you. It's a matter of you being able to keep up with those threats, which is why evolution continually tries to keep up with the threats of predators getting better and better, or your prey getting harder and harder to find, or the bacteria or the viruses, or the prions, you name it, fungi. So red queen theory really is sort of at the essence of this. What's going on with aging is not that entropy wins, but that maintenance loses. Entropy wins in a sense because maintenance is turned down. Now, the other interesting question is sort of a teleologic one, which is why does maintenance get turned down? And it turns out there's some interesting evolutionary answers to that, why we age. And again, as you might expect, it's even more complicated than you first think. But aging is not a matter of entropy, it's a matter of maintenance. And maintenance turns out to be modulated by these changes in gene expression that are in turn modulated by changes in the length of telomeres that are modulated by cell division that in turn are modulated by other things. For example, the degree to which I undergo trauma or infection, all of which increases or if I smoke or drink a lot or anything, those things will increase my rate of aging. If I go out and get sunburned a lot, I increase skin aging. But the problem is, as I try to replace damaged cells, I shorten telomeres, change telomere length, change gene expression, change maintenance, turn down protein turnover and recycling, turn down DNA repair. And the next thing you know, I'm old. Yes, it's complicated, but it's in principle, the concepts are startlingly obvious.
And if
in retrospect.
If I understood correctly, and I will take it like to the next level, then let's say even if we manage to like break the 120 limit, as we know today, of aging, and we knew what are the mechanism we need and how to maintain them. After we'll break it, part of evolution is that we will need to deal with maybe another
All right, I think I see where you're going. So let me give you two examples of this. Let's just look at the lungs for a minute. And let's take two very young people with excellent lung function. And one of them gets a series of lung infections, pneumonias, for example, bronchitis, anything that involves anything in the pulmonary system. Now, that person who has a series of infections will have early aging of the lungs because you're turning over, for example, the pneumocytes, for example, the fibroblasts, for example, the immune system that's involved with lung function and the vascular system. You know, there've been injuries, cells being replaced, it ages faster. So this person will end up with early lung disease, for example, COPD or pulmonary fibrosis, okay? Now, that kind of problem could be dealt with telomerase. I can reset the pattern of gene expression and give them theoretically young lung function. Now let's look at a different case. Here's a person over here, started out with identical lungs, very healthy, never had any infections, but they work in a mine and they're continually exposed to silica particles of all sorts. And they begin to get depositions of silica particles throughout the lung. This person, that's not an age-related process that I can address with telomerase at all. The fact is those silica particles are causing damage to the lung just by being there, and I can't get them out. It's simply a matter of accumulated damage. Whereas these ones, we could reset, those ones we can't. So, you know, maybe another example might be no, let's leave it at that. That's probably the best example I can come up with instantly. It's some things are age-related, some things are just related to the fact that you've been sitting on this planet too long.
Yeah. Okay, so well, I will ask like a general question about Alzheimer. When you say we might reverse it, what do you mean? That we will get memories back that will last, or how will it look like?
Years ago, some 24 years ago, I gave the first talk ever at the National Institute of Health in Bethesda, Maryland, on aging and the possibility of reversing it. And one of the things I said was that anyone who leaves this talk in an hour and thinks you can reverse aging is naive. Anyone who leaves this talk in an hour and thinks you cannot reverse aging is naive. The question really is, where's the data? And that's kind of what's going on here too. Now, let me give you some of what we do know. We do know that we can actually improve the behavior of animals. That is, almost any animal shows behavioral decline with age. You know, what is it? You can't teach an old dog new tricks. Well, if we look at animals that we try treating this with these mechanisms that we're going to take to human trials, you find you can improve their ability to learn, their ability to do behavioral things that they weren't competent at before you treated them. So we know we can get return in many ways. We also know there's evidence that in Alzheimer's, much of the loss, not clear how much, but much of the loss is not a loss of memory, but your ability to access the memory. That is, the memory's there, you just can't get at it. Now, let me add another piece to this. If I take any patient with Alzheimer's, the odds are that there are a number of cells that are damaged but still functional, and there are a number of cells that are gone. They're just gone. And I probably can't get back cells that are gone, let alone reestablish the right connections. But if I take cells that are damaged but still functional, I can probably make them more functional. What that suggests, what the data suggests from animals, from humans, and from what I just said, suggests that we probably can get improvement in Alzheimer's patients, but not everything. Okay, so my guess is something like this. Let's take a scale, and I'm going to make up this scale from 0 to 10. There are many different Alzheimer's scales, but let's just imagine one from 0 to 10. And 0 is I have no problem, and 10 is I'm at the very end of my life in the nursing home, and I can't care for myself, barely breathe. My guess is that if we take patients with about 4 or 5 on that scale, we can get them back to about a 2 or 3, rather than a 0, rather than not being able to affect them at all. I just don't know the limits of that. Back to my remark at the NIH, which is, show me the data. Let's see what happens.
Okay. And where are you, like, regarding to the phase 1?
Well, the biggest problem, you know, first, obviously, there's a problem of getting the financing we need to do this. And that involves convincing people that it's worth trying in the face of all prior failures and having an innovative model for how these things work. The second is the time it takes to produce them. And the third is actually.
1 second, but this model still didn't been tried.
I'm sorry, what?
This model wasn't ever tried, like as
not in humans yet, no.
In humans, yeah.
No, in animals, yes, in humans, no. So, you know, it's an innovative model. And as I say, everything else has failed. They sometimes say that Alzheimer's is a graveyard of companies, okay? Or that everything works in mice, nothing works in humans. So there are a lot of caveats. But so one, convincing the investors to move ahead. Two, the FDA has to sign on to what we want to do. And three, we have to have the vendors produce in quality and quantity what we need for this. So there are sort of rate limiting steps to moving ahead with all this. Our best estimate is it will be about 2 years before we start the first safe human trial, and that we'll have results within 3 years.
Again, there was a disconnection. You have a few steps, and then it connected.
Yeah, the first step is making sure we have the financing actually in the bank. The second is convincing the FDA.
You hear me?
I lose you again, Joe?
Yeah, I'm losing it. Well, we are far away from each other. So. Yeah, now it's working clean.
The first step is the financing. The second step is the FDA. And the third step is having the vendors produce the quality and the quantity we need to do this safely and effectively.
Yeah. And if I understand the biology correctly, like every cell has a chromosome, and it has telomeres in the end.
Every cell has what, Gil?
Every cell has a chromosome with a telomere in the end of it.
Mm-hmm.
And so we can have a lot of diseases that are related to this kind of problem.
Mm-hmm.
I'm correct?
Yes. You know, one of these questions is, how come you get Parkinson's and I get Alzheimer's, or I get Alzheimer's and you get coronary artery disease?
No, the question is, why not to start with trying to cure a disease that is related to an organ that is not in the brain with the same method?
That's a question that we sort of addressed before. It has to do with the risk benefit. You know, I could go after osteoarthritis, but right now, the standard treatment for osteoarthritis is, for example, non-steroidal anti-inflammatory medicines or replacement of the joint. So there are options, and osteoarthritis doesn't kill people. Alzheimer's is a better target because it's fatal and there's no other treatment. So it's a much more effective target for us.
Yeah, I understand. Okay. So, well, we covered a lot. We covered almost everything. Let me ask you, if I was a genie, I asked Dr. Brian Kennedy the same question, for the former CEO of the Buck Institute.
Yes,
if I was a genie, yeah, what is your 3 requests to make it happen faster?
Not just faster, but correctly. You know, my biggest worry is not that the model is wrong, but that we will not test it carefully enough. That is, we'll do something wrong in trying to get this to work right. Back to the example of the Wright brothers. There are lots of ways to fail getting their first plane off the ground. But they were right, but they needed to get it exactly right to get the plane off the ground. That's sort of where we are with regard to gene therapy these days. Everybody thinks it's simple in principle, and in principle it is, but the technical details are remarkably hard. Example, you know, in the US, about 60 or 70% of people already have antibodies to the sort of virus I want to use. So can we give it effectively? There are some risks to damage to certain ganglia in the spine if we use AAV, and yet we can get around both of those problems, but we have to be careful how we do it. We also know that the kind of approach we're using doesn't give us the efficacy we'd like to have. So can we improve it? Yes, we can. But what are those ways of improving it are the safest and most effective? This is early, and it's much more complicated than most people think of it as.
Yeah.
So yeah, that's my genie wish. I would like to get this right. I'd like to save lives and make people have better lives.
Okay. How does the FDA? You said like you're working with the FDA needs to prepare to gene therapies to do their own um, process. Are they doing something about it? Like it's a new concept?
Yeah, it's new. I mean, it's worldwide. It's new. It's been around a while, and of course, there were some setbacks back almost 20 years ago with some early deaths. Jesse Gelsinger comes to mind, and that made us all pause and be very careful for a decade or so. And there's still some issues coming up. So if I look at the FDA, basically you can think of it as being two sections. There's the CEDAR section, which looks at small molecules, and there's a CBER section that looks at biological treatments like gene therapy. And this has been well known for a long time. The processes are well understood. This is new, and it is changing, in a sense, month to month. And the people involved in this at the level of the FDA and CBER are very bright and very enthusiastic and eager to see things work, but they don't want to make it, they don't want to screw it up either. They want to make sure it's done carefully. And I can't but agree more. I entirely with them on this. But the consequence is that some of these, the things we do and how we do that safely are unknown, they're changing. Example, if I give you a gene therapy, how long do I need to follow you? Because you may still have the virus. Do I follow you for a year, 6 years, 15 years, forever? Do you shed the virus? Can you infect other people? Again, what are long-term risks? What about risks of cancer? All of these things are in flux in some sense. And, we've got a lot of people know a great deal about it, but not as much as we need to know. And I think that CBER and the FDA and the EMA in Europe and others recognize that. And I think anybody involved in gene therapy recognizes that. So we're doing the best we can.
Yeah. And like in terms of sorry, I lost my. No, I had a question, but I waited and. Yeah, it was a question that was referring to what you said, and I just lost. You see, we're talking about memory, and I'm young, but yeah, that's an example.
Funny thing is I often have said that, if at age 20 you forget your car keys, you don't worry about it. At age 70, you forget your car keys and you assume you've got Alzheimer's. The older you get, the more you worry about these things, even though everybody forgets things.
Yeah, everyone. Well, I was in one lecture and they said if you forget what the kids needs to do, then you need to start worry.
But how many times have you and I and everybody had a time where you walked into the other room to get something, got there and can't remember what you were going to go get? That's welcome to life.
Yes, we need not to take it too seriously because it just.
Well, but like so many other things, if you have a chest pain at age 20, you don't think much about it. If you have a chest pain at age 60, you think you've got a heart attack, and the odds go up, or a lump, a cancer. Yeah. Odds go up with age, and yet it's sort of part of life to have lumps and pains and memory problems. It's not new for any of us.
Yeah. So, are you like in New York company, or are you sitting in San Francisco?
No, a typical virtual company. I am currently right now in Michigan. Our CEO in the United States, our CEO is in the UK, our COO is in Boston, our chief consultant is in Scotland. Our attorneys are all over the world. They joke that they really live at 35,000 feet in aluminum tubes. And our chief vendor for production is really in Texas. Our first medical site will probably be in Kansas, although we're looking at several other possible sites. And then our phase 2 trials will be global. So it's typical of a company in these days, particularly in these days of COVID.
Yeah.
It's like I didn't go to the Amsterdam conference this year. I'm not going to the conference on gene therapy this week. I'm following it online.
Everything is online. Yeah.
Pretty much. And our investors said the same thing. The first question was, do you have brick and mortar, which is, do you have a building? And we said we didn't. They said, good, we don't invest in brick and mortar. Times have changed.
Yeah, that's actually, if we're talking about investors, it's quite reminds me that the question I forgot, like how much like the investment society are willing to invest in gene therapies these days.
I'd say that well less than 1% of the people that you get in touch with regarding investments are actually wanting to follow through and invest. I'm not sure how atypical that is. And I know that the biotech market has done very well over the past 11 months or so with COVID. But it's part of the problem, of course, is that people would like to make a lot of money but not have any risk. Doesn't happen. People are not willing to invest in innovative things in general, even though they often say they are. And people are often willing to invest in things that they're familiar with. So some companies only want to invest in software, some only in medical devices, some in diagnostics, some in innovative therapeutic approaches. So it's hard to find just that right match. We have found the people who are willing to invest in us, and we'll probably move ahead very quickly now.
Yeah, and if you're speaking about diagnostic, like you can reverse, like you want to reverse aging, and I know a lot of companies are trying to build like diagnostic that will predict if you will have disease, what kind of disease you will have, and will try to prevent. But can we refer to your treatment as a preventing the, therapy too?
Almost certainly yes, but think about how difficult it is to prove that. So let's say that I don't, but let's say that right now I have a shot right here I can give you that will cure your Alzheimer's. Let's say that's true. How would I now prove, and I can prove that, like you have Alzheimer's and I give it to you and you get better. Let's say that's true. Now, let's say that you could also use the same thing to prevent Alzheimer's. How do you prove that? Well, what you have to do is find thousands of patients who will get Alzheimer's in the next 10 years, give it to them and wait 10 years and compare to the people who didn't get the shot. You know how difficult that is? For one, that takes 10 years. And two, if there's any evidence that it works to cure it, nobody wants to get the placebo. So how do you prove it prevents Alzheimer's? The same is true of many other diseases. If you're trying to prove these things, it's very difficult for prevention, whereas intervention might be easier, even though most of us would prefer prevention in the first place.
Yeah. And how is the participant, like people want to participate in Alzheimer trials?
Yes and no. For us, yes, because we're looking for patients with moderate degrees of Alzheimer's. The ideal patient, I've been on some of the global patient recruitment calls, and the ideal patient for most companies worldwide, both little biotech and big pharma, is the patient who hasn't got Alzheimer's, but will get it quickly. That is, they've got gene risks, they've got known laboratory risks, they've got family risks, they've got other risks that predict that they'll get Alzheimer's, but they have no symptoms right now. And the rationale for that decision, and most companies are fairly honest about it, most companies will tell you they don't feel that they can stop Alzheimer's, but if they catch it very early, they could maybe slow it. Think of it as catching that first couple of pebbles in an avalanche. And the consequence is they're having a very hard time finding patients because their perfect patient not only would have those characteristics, but would also have MRI scans and PET scans and data that show that things are about to happen, which are expensive. So they're fighting for these patients, and it's very hard to find them. We're actually looking for patients with moderate Alzheimer's because we think we can actually improve cognitive function. So it's a lot easier to find patients who want to volunteer who already have Alzheimer's than it is to find patients who don't have it but might get it. So it's very easy for us, very hard for most companies.
Yeah. So this is one point, like a good point for Alzheimer for your company, because in cancer, I know it's hard too. There's a lot of competition on the patients.
Very much so. Yeah.
So maybe
this year with COVID, when no one wants to go in and be in touch with other people and get COVID as well as cancer. So it's a very tough year for many clinical trials.
Yeah, I think Israel has bought from two companies, from Pfizer and Moderna, The government, like, will is supposed to give it, but still people are afraid to take it.
Vaccine, you mean?
Yeah, vaccine. Yeah.
I understand. I'd be eager to take the vaccine, but I understand some people have qualms about it. Okay, I understand.
Yeah. So it still doesn't solve like completely the problem, even if we will have it. And okay, Michael, thank you again for your time. Again, you are. I saw a lot of your content podcasts, and each time you give 100% of yourself, and I appreciate it.
Tada.
You're welcome. Bye. Okay.
להתראות,